What is meant by histology? The study of normal tissues and its structure. Histopathology, the study of tissues in abnormal and cancerous condition. The histopathology is divided into two types, biopsy and autopsy. Biopsy is a procedure to remove a place of tissues or sample of cells from your body so that it can be tested in a laboratory. Autopsy means to determine the causes and manner of death as well as to evaluate any disease or injury that may be present. Tissues preparation. Examining the tissue samples through the microscope is not as simple as cutting slices and looking through the lens. There are several steps that must be taken at the sample acquisition before a sample is ready for the scope. There are four major steps for processing and preparation of tissues: fixation, dehydration, embedding, and staining. Fixation. What is meant by fixation? To preserve the tissue structure in the original condition, and it is a morphological structure is not particularly proper arrangement. It is called as fixation. The fixation is used for prevent self-digestion, kill foreign material such as bacteria or viruses, harden the sample as a result of cross-linking and protein denaturation, or body organs are highly ordered, very precisely structured. Fixative reagents: formaldehyde, acetic acid, ethanol, osmium tetroxide, and chloroform, toluene, toluol, etc. Next one, decalcification methods. What is meant by decalcification? The removal of calcium from bone and tissues. It is called as decalcification. The decalcification is used for complete removal of calcium salts, minimal disorientation of cell morphology, no interface during staining. Therefore, in order to attain such precision and safety and satisfy the above condition as a successful process of decalcification, this follows is required a careful preliminary assessment of the specimen through the fixation, preparation of slices, reasonable thickness of our fixation and processing. This choice is suitable and decalcifier with accurate and adequate volume changed regularly. A careful determination of the end point through processing using a suitable schedule. This decalcification is mostly three methods: acid decalcification and minimal methods. It is commonly used for the method for acid decalcification. Reagents required for acid decalcification: nitric acid and formaldehyde composition. Nitric acid is 10 ml and formalin 5 to 10 ml, and make it to 100 ml. Procedure: it has the following advantages: rapid action, good nuclear staining, and washing with water is not required. Next one, principle for tissue processing. The technique of getting fixed tissue is into paraffin wax is called tissue processing. Tissue processing is designed to remove all extractable water from the tissue, replace it with a support medium that provides sufficient rigidity to enable sectioning of the tissue without damage of distortion, act as influencing tissue processing. And there are several steps: agitation, heat, viscosity, and vacuum. Agitation: it increases the flow of fresh solutions around the tissue. Heat: it increases the rate of penetration and fluid exchange, and temperature limited up to 45 degrees Celsius can be used. Temperature higher than that the causes of tissue brightness. Viscosity: it is a property of resistance to flow of a fluid. Vacuum: using pressure to increase the rate of infiltration decreases the processing time. Next one, step processing. Second one, dehydration. Dehydration: clearing and embedding. What is meant by dehydration? The removal of water from the tissues present the fixative components. Many dehydrating agents are hydrophilic and interact with the water molecules in the tissue by hydrogen bonding. Other reagents affect dehydration by repeated dilution of the aqueous tissue fluids. Dehydration should be accomplished slowly. Dehydrating agents, ethanol, methanol, and methylated spirit, isopropanol and isopropyl alcohol, normal and tertiary butanols, dioxin, polyethylene glycols, acetone, phenol, anhydrous copper sulfate, HCl, and tetrahydrofuran. Next one, clearing. Clearing is the transition and intermediate step between the dehydration and the embedding. Selection of clearing agents, rapid penetration of tissues, rapid removal of dehydrating agent, easy to remove of melted paraffin wax, minimal tissue damage and low flammability and low toxicity and low cost. What are the clearing agents? Toluene, toluol, xylene, chloroform, carbon tetrachloride and trichloroethane and esters, etc. This commonly is for xylene. Xylene, it is a flammable and colorless liquid with a character petroleum odor visible with the organic solvent and paraffin wax. Overexposure may cause tissue hardness. Mostly commonly used for clearing agent in routine histology and is also used. It is a recyclable process. Toluene, it is the same properties as xylene. It is damaging with a prolonged immersion of tissues. It is more flammable and volatile than xylene. Next one, chloroform, slower in action and a little brightness. Thicker blocks may be up to 1 mm in thickness can be processed. Next one, embedding. It is a process by which tissues are surrounded by a medium such as agar, gelatin and or wax. Properties of embedding media. Ideally, an infiltration or infiltrating and embedding medium should be soluble in processing fluids suitable for sectioning of ribbing and molded between 30 degrees Celsius to 60 degrees Celsius. Translucent or transparent color, and it is also colorless and stable and homogeneous, and it is capable of flattening and after ribbing. These are the embedding process. Next one, paraffin wax. Most popular embedding medium in histopathology. It is a mixture of long chain hydrocarbons produced by the cracking of mineral oils. Paraffin wax permeates the tissue in a liquid from a solidifier rapid cold. Next one, types of paraffin wax. Picolate 115 and plastic polymers and dimethyl sulfoxide. These are the paraffin waxes used for histopathology. Embedding tissues in paraffin wax required for embedding as follows. Following requirements. It is a supply of clean filtered paraffin wax held at 2 to 4 degrees Celsius above its melting point. It is a paraffin dispenser, and it is a required cold plate or rapidly cold and wax. A supply of molds in which embedded in their tissues. Paraffin wax is dispensed automatically from nozzle into a suitable sized mold. The tissue is oriented in the mold. A casting is attached. The mold is placed on a small cooling area to allow the paraffin. Reagents. Procedure for embedding. 10% formalin for 1 hour at 30 degrees Celsius, and after dipped wash in water and 50 degrees Celsius, 50% alcohol and for formalin dipped at 1 hour and melting temperature at 30 degrees Celsius. Next one, 70% alcohol dipped with 1 hour and maintained for 38 degrees Celsius, and 95% alcohol dipped with 1 hour and maintained for 30 degrees Celsius. Next, 100% alcohol dipped in with 1 hour and after take maintained for 88 degrees Celsius. Next one, xylene with 1 hour and maintained for 30 degrees Celsius. The same also xylene again dipped 30 minutes and maintained for 30 degrees Celsius. Next one, microtom or sectioning. What is meant by sectioning? Microtom, it is an instrument. It is used for sectioning, cutting in that technique of making a very thin slices of tissues specimens for the microscopic examination to identify the abnormalities or atypical appearance in the tissues, and also for the study of various components of the cells or tissues like lipids, enzymes, antigens, antibodies, and immunohistochemistry and other cell organelles, etc. Before sectioning or made, the tissues required to be embedded in some fluid which of the permeate these sites and is capable of being rendered from so as to support to the most delicate parts of the knife passes through the tissue. The most commonly used for embedding medium in paraffin wax as it is well allow optimal sample preparation. Next, after the process, deaeration, clearing, decalcification, etc. Types of microtomes. There are 4 types of microtomes: racking microtome, random microtome, freezing microtome, and Cambridge microtomes. Next one, hematocylin and eosin staining principle, procedure and interpretation. Hematocylin and eosin staining is the most widely used staining technique in histopathology. It is as the name suggests. He stain makes use a combination of 2 dyes, namely hematocylin and eosin. This combination differentially stains various tissue elements and make them easy for observation. Principle. The hematocylin eosin stain is the chemical attraction between tissue and dye. Hematocylin is a basic dye, imparts blue purple contrast on basophilic structure, primarily those containing nucleic acid and moieties such as chromatin, ribosomes, and cytoplasmic regions such as and rich in RNA. And acidic eosin counterstains the basic elements such as RBCs, cytoplasm, muscles, and collagen in varying intensities, pink and orange, red. Reagents required for hematocylin staining. Hematocylin 1 gram in 10 ml ethanol and B for 20 gram for ammonium aluminum in hot distilled water and mix A and B solution. Next one, eosin solution. Yellow eosin 1 gram and distilled water 580 ml and ethanol 320 ml. Glacial acetic acid for 2 drops and mix well 0.5% HCl and ammonia water. Next one, procedure. First, deparaffinization from the flame the slide or on burner and place in the silane. Repeat the treatment to remove the wax. And the next one, hydration. Drain xylene and hydrate the tissues, sectioning by passing through decreasing concentration of alcohol bars 70% alcohol and 80% alcohol and 90% alcohol and 100% alcohol. Each one dipped and washed in running water with 2 minutes. Next one, nuclear staining or primary staining. It is a hematocylin staining is added 3 to 5 minutes wait and after wash in running water. Next one, differentiation. Selective removal of excess dyes from the sectioning. Dips in 1% acid alcohol and wait for 5 seconds and wash in running water. And the next one, bluing. Rinse in running tap water dipped in ammonia water until the sectioning becomes blue, followed by tap water and wash in running water. Next one, counter staining or mounting stain. This stain is added and 1% eosin stain. It is present in yellow color and wait for 10 minutes and wash in tap in running water with 1 to 5 minutes. Next one, dehydration. Dehydrate in increasing concentration of alcohols and clearing, put slides into 2 silent bars for clearing. Mounting. Mount in DPX or other mounting media. And finally, seen under above the microscope with 10x and 45x and 100x of oil immersion. Results. Blue or black color, it is appearance which is present in nucleus, and a pink or bluish pink color is appearance. It is appearance for cytoplasm. Deep red color is present in it is appearance for muscle fibers. Orange red color appearance which result from RBC cells. Dark blue color is present which result is called as calcium, and grape blue color is present which is called as mucin and other cell internal organelles.